Startovali chvilinku. Tak jo, myslím, že to můžeme připustit. Pavle, začneme.
Good afternoon, everybody. It is my great pleasure to introduce our candidate for associate professorship at Masaryk University, Dr. Pavel Babica. Reading his CV, I found out that we have quite a few things in common. We were born in the same region, I found out. I think we even joined Recetos at the same time because I remember the year when I joined, you were the bachelor student I was teaching in Pavel. And we also both spent 5 years at Michigan State with many other people in the room as well. Pavel, he's been with us for many years. He started as a bachelor student and then master and PhD student, co-supervised by Professor Marchalik and Professor Blaha during his studies. After that, he left and he spent between 2006 and 2010 in East Lansing working in Michigan State University as a postdoc. Coming back, he worked at the Institute of Botany of the Czech Academy of Science, where, if I'm not mistaken, he still has a part-time job. And then since 2012, he returned back to Masaryk University and is again working at the center. During his career, he published more than 40 papers. He supervised and co-supervised 10 different PhD theses and a number of master and bachelor theses. And the name of this presentation today is Cat Counting Intercellular Communication in Vitro, Assessment of Hazardous and Beneficial Effects of Chemicals. So thank you for coming, and floor is yours.
Okay, thank you very much, Sir Klanova, for the very nice introduction, and also I would like to thank all of you for coming, and also good afternoon to those of you who are watching this online stream. So, when I was asked to prepare this lecture, which is part of this habilitation procedure, the topic was quite an easy choice because I spent a large portion of my research focusing on the role of junction intercellular communication and its use in toxicological or pharmacological assessment of chemicals. And I try to basically summarize an outline of this lecture in this cartoon, or let's say graphical abstract. So what is it going to be about? So cells in our body, they usually talk to each other. They like to talk a lot. And one way of communication between them is actually mediated through this, let's say, prehistoric but still very functional type of cell phone. And so they talk to each other, and sometimes they see something strange in the environment, so they may freak out, they can change the communication, and maybe they stop talking at all. So we, as a scientist, we can use good old spying, and we can actually listen to what they are talking about, and from that we can actually realize how they feel about the environment, and for example about the presence of some chemicals which we would like to know more about. Modern toxicology and pharmacology, why that? So basically modern toxicology and pharmacology are important societal need of modern civilization, which has been very successful in producing and releasing many different chemicals into the environment. Some of them are produced accidentally, some of them are produced intentionally. Some chemicals we are actually using on purpose, for example, drug use or abuse. So, of course, we would like to know what might be the effects of this continuous exposure to different types of chemicals. What might be the health effect or impact on our health. Of course, some of the chemicals, they can be very beneficial for us, so they can improve our health condition, and we can actually take advantage of that and use these chemicals, for example, to cure or treat different types of diseases. Some other chemicals might be very dangerous, toxic, they can actually have adverse effects on our health, and it's better for us not to come into any contact with them, or at least minimize the contact. So basically, we would like to know which chemicals are the good, which are the bad, and which are the ugly ones. And for that, we can use the modern pharmacology and drug discovery techniques, or on the other hand, modern toxicology and toxicity testing to figure out who is who, basically. When we would like to know, let's say in advance, or predict what might be the effects of chemicals which haven't been even started to be released into the environment, or haven't been started to produce. So for that kind of predictions, we can use tools or methods of experimental toxicology and pharmacology, basically to discover adverse health effects or predict them, or to predict therapeutic effects and potential of different drugs. Modern toxicology and pharmacology has been traditionally based on studying organismal response using different type of in vivo assays. However, it looks like that in the recent years, the tables are kind of turning in favor of using in vitro or in chemical or in silico, let's say animal-free methods. There are many different reasons behind that, but basically this shift has been partially driven by recent advances in different technologies and science. For example, cell culture techniques and tissue engineering now allows to work in vitro with different well-characterized cell lines, which are representing different types of organs, tissues, developmental stages, using three-dimensional cell culture techniques. We can actually mimic physiological or in vivo-like conditions in these in vitro systems. Advances in molecular biology, microscopical techniques, bioanalytical techniques, biochemical techniques now allow us to study in detail the underlying mechanisms of the interactions of chemicals with biological systems. There has been great development in so-called omics techniques. So nowadays, instead of studying one gene, protein, or endpoint, we can study tens, hundreds, or thousands then. We have also bioinformatic apparatus which can process these large types of data. And of course, progress in robotization and miniaturization allows to do these in vitro experiments in high throughput setups, so we can really acquire a lot of data about many chemicals in vitro. So in today's talk, sorry, I think I skipped one. Oh, it's not there. Never mind. Yeah, so in today's talk, I would like to actually talk about gap junction intercell communication and what is its place within these recent developments in toxicology and pharmacology and methods. So, what is cell-cell communication? As I already implied in the title of this lecture, it's evolutionary, very old or ancient type of biological process. So here you can see the phylogeny of life plotted across the time. So we can see that around 4 billion years ago, life has evolved on the Earth, and for the vast majority of time, for up to I think 3 billion years, it remained in this prokaryotic form, which is characterized by existence of isolated cells or maybe colonies, but they are usually colonies of one cell type. So these prokaryotic organisms, they do not form complex multicellular bodies like eukaryotes, which evolved about 800 million years ago. So it really took 3 billion years to get from this single cell life to this complex multicellular life. Evolution of multicellular life is associated with evolution of eukaryotic cells, and you can see that once that happened, so we can see really explosion of complex multicellular eukaryotic life, which is actually characterized that the eukaryotic organisms, the multicellular ones, their bodies, they are formed, like in the case of humans, from up to several hundreds of different cell types, and our bodies are made from several trillions of cells. So that's important difference between these two types of cell life. And it really seems that there has to be some critical condition what happened during the evolution, which actually allowed this explosion. So if we are thinking about what might be critical for existence of multicellular life in the form we know it, we can think about the individual ontogenic development of organisms, because although we as adults exist in this 40 trillion cell human bodies, 200 different cell types, life of every one of us has begun from a single cell which was fertilized egg, and somehow from this single cell we managed to get into this proper form of adult human being. And I think, okay. And this actually happens through this strictly regulated and organized process of individual development. So this fertilized cell, so fertilized egg, is able to proliferate, and along the development, the progeny of these cells actually starts to differentiate into more specialized cell types, and finally to terminally differentiated cells which form our tissues and organs, and that's how we become this 200 cell type, 40 trillion cell human beings. So regulation of these processes actually depends on three critical conditions. So there must be in place mechanisms which control the growth of these cells, so they do not just proliferate infinitely, as they actually can, but sometimes they know where to stop proliferating. So that's how we end up with a proper number of cells which are needed for our bodies. Another important feature is the induction of specialized cell types, so differentiation, which is actually mediated through regulated expression of the selected sets of total genomic information. And the third important feature is programmed cell death. So sometimes, or actually all the time throughout our life, we have to remove cells which are either damaged or no longer required or functional. So they can be replaced with functional and proper cells. And these processes are regulated through dynamic interactions between genes and proteins and their environment, or in this case, if we are talking about the development microenvironment, so where the cells is placed, so it receives important signals from its microenvironment or neighborhood, and these signals actually modulate the expression of genetic information. So as you know, pretty much all the cells in our body, they have the same genomic information, so there is not much difference in that. But based on these signals from the microenvironment and this interaction between genes, proteins, and environment, the cells are able to turn on and off different genes. And basically, in response to that, they can modify their behavior, so they can either proliferate, they can differentiate, or they can, for example, undergo apoptosis. So basically, cell signaling kind of mediates the formation of this epigenetic landscape through the cells can actually move along the desired outcome. So that's a healthy human being. So if we are talking about cell communication, which really is important for the modulation of gene expression and all these developmental processes, there are basically four types of cell communication. So cells may respond to extracellular stimuli, so some secretory molecules which is released and then binds to some kind of receptor. Cells also interact with the extracellular matrix through different types of proteins. Also, there might be ligand receptor interactions between cells. And last but not least, there is the symplastic type of communication or symplastic connections between the adjacent cell within a tissue. Through these connections, the cells can actually exchange important signaling molecules and sort of coordinate their behavior and responses across the tissue, and also integrate all kind of signals which the cells are receiving. And it turns out that this type of communication is probably very important for the existence of multicellular eukaryotic organisms, because in all major groups of eukaryotes we can find some type of direct cell-to-cell communication. So in green plants and brown algae, you can find these plasmodesmata, so the cells are really connected through physical channels, so they can communicate through them. In red algae, there are these pit plugs or pit connections. In fungi, there are septal pores, and finally in animals or metazoans, there are gap junctions which are actually mediating this type of communication between the cells within our tissues. So, cell-cell communication in vertebrates, as I said, is mediated through these so-called gap junctional channels, and the process of exchange of soluble molecules through these channels is called the gap junction intercellular communication. These channels are built from proteins called connexins. In human genome, we can find 21 different connexin genes, and these genes or proteins coded by these genes, they create within or in the plasma membrane of our cells these hexameric structures, or structures which are called hemichannels or connexons, and two adjacent cells can place these hemichannels against each other, and then they can start exchanging important metabolites and molecules between each other. So the type of molecules which can pass through gap junction intercellular channels, it includes mostly small molecules, smaller than 1.5 kilodalton usually. These molecules include common metabolic products like glucose, glutamate or ATP, but they include also important secondary messengers or modulators of signal transduction pathways, such as cyclic AMP, cyclic GMP, inositol triphosphate, glutathione. They can exchange also ions, reactive oxygen species, and recently it has been published that actually cells exchange microRNA through these channels. So basically, as I said, this communication allows the cells to kind of behave in a coordinated way to respond to different stimuli, not as a bunch of individuals, but they can, as I said, coordinate their responses and integrate different type of signals and stimuli from the surrounding environment, and through this mechanism control the gene expression and cell behavior. So, since this process is really critical, so the timely regulation of cell-cell communication is necessary. So the cells need to communicate in in the right time in a right manner. So there are different mechanisms which can regulate this type of communication. So on this picture, you can see modulation of sensor communication through so-called gating mechanism. So that is really some physical-chemical change of the connexins within the membrane. In this case, it's a phosphorylation of C-terminals, which kind of makes conformational change of these C-terminals, and that clogs the channel, so the metabolites or signaling molecules cannot be exchanged anymore. Another example is modulation through the calcium, which can bind to this part of connexin molecules, and again there is a conformational change which really physically blocks the channel. And here are some microscopical pictures of the process. So here you can see the channels, which are usually grouped in so-called gap junction plaques within the plasma membrane. So there are not single connexin molecules, but usually tens or hundreds of them organized in the membrane. And you can see that this is the open channel, and here upon the calcium treatment, you can see the closure of the channel. Of course, this is just one mechanism of regulation of sensor communication. It can be also regulated on the level of expression of different connexin genes. As I said, there are 21 of them in the human genome, and not all of them are always expressed. There are modulations of translation and maturation of connexin molecules. They're docking into the plasma membrane. The connexin molecules have a very short life, so usually the time from the protein synthesis till the degradation is between 1 hour to 20 hours. So there is very rapid turnover of connexins within the cell. So any process which somehow interferes with the gene expression of connexins or its proper placement into the plasma membrane can very quickly lead to the modulation of sensor communication. As I said, it's an important process, so no wonder that it's very connected and regulated to the whole signaling network, this signal transduction pathways. Actually, connexin is phosphoprotein, so this C-terminus, which is in the intracellular space, can be directly phosphorylated by the major signal transduction enzymes or kinases, such as SAR kinase, nitrogen-activated protein kinase C, and many others. Connexin molecules also interact with cytoskeletal proteins, and through that cytoskeletal apparatus also with other types of junctions. And again, modulation of these interactions can result into the change of channel permeability and modulation of cell-cell communication. So why is this communication important? So I did already mention that they are involved in the growth control, regulation of apoptosis, differentiation, metabolic support. In some tissues, they have really special function, like in the cardiomyocyte, they mediate these electrical synapses. Also, they are important in the tissue regeneration and maintenance of tissue homeostasis. What happens if the connexins are not really working very well? So there have been some experiments with transgenic mice models. So we can see that knockout of some connexin genes is lethal, either embryonic or early life stage lethality. Some other connexins, they can be, they are knocked out, they can be partially or nearly fully compensated, but it's usually associated with different types of diseases or pathologies. So in human beings, there have been several congenital diseases linked to the mutation in connexin genes, for example, cataracts, microphthalmia, peripheral neuropathy, hereditary deafness, also cardiac arrhythmias. All this has been linked to mutation of some of the 21 connexin genes in human genome. Moreover, if you look pretty much into any chronic disease or pathology, you can find that the cell-cell communication is not working as it's supposed to do in normal healthy tissue. So it includes nervous system diseases, neurodegenerative diseases, for example, cardiovascular diseases, pulmonary, digestive system diseases, all kind of inflammatory diseases, reproductive, and also cancer. So, how might connection channels or cell-cell communication respond to the exposure to different type of chemicals, either let's say endogenous metabolites or some chemicals which are coming from the outside environment. So some molecules have been really clearly linked or shown to stimulate or promote cell-cell communication, and it's oversimplification, but let's say these are like good chemicals, or it's rather desired that the cells in our body communicate. So that includes inhibitors of growth, compounds which induce differentiation and are also epigenetic modulators like inhibitors of histone deacetylases, also some anti-cancer drugs or chemicals which have been linked to the cancer chemoprevention. On the other hand, chemicals like tumor promoters, growth factors, inflammatory cytokines, and all kinds of environmental toxicants, starting from organ-forming pesticides through fluorinated fatty acids, phthalates, to metals, have been found to inhibit or disrupt this vital biological process. So that looks like that by evaluating junction intercell communication, we can actually use this biological process as a biomarker to assess potential effects to either disrupt or restore this critical biological phenomenon, and that can give us some indication about the potential of that chemical either to induce or treat chronic toxicity or some diseased states. So to be able to do that, and as I said, preferably using some in vitro assays, we should be able to measure cell-cell communication in vitro conditions. So for that, there have been several different and principally different methods developed, and what I'll be presenting in the next slides, it's a method which is used in our laboratory, and it's so-called scalpel loading dye transfer technique, which is basically based on the introduction of fluorescent dye into the cell monolayer by a cut with a scalpel blade, and if the channels are open, so you can see that the fluorescent dye can travel from the place of the cut into the neighboring cells. On the other hand, if the communication is inhibited, so then you don't see any dye transfer at all. So that's a very simple technique. Also, if you would like to use this evaluation of cell-cell communication more often in toxicological or pharmacological research, it would be really desirable if we can measure this effect in different types of cells representing different organs or tissues. So this is just a collection of examples of different cell lines where you can effectively evaluate gap junction intercell communication. So you can see it includes liver cell lines, human and rodent, some cell lines from respiratory system, intestinal cell lines, somatic testicular cells, or neural cell lines. So that can be measured in different systems and setups. Also, when I was listing all the different chemicals which can actually inhibit cell-cell communication, so it may look that pretty much anything can inhibit cell-cell communication, so that wouldn't make much sense to further study this process. But actually the effects of chemicals on cell-cell communication are very structure specific. So here you can see some examples of pairs of compounds which are structurally similar, but they have very different effect on the cell-cell communication. So the blue ones, they do not inhibit cell-cell communication at all. The other ones, the red, they are potent inhibitors of cell-cell communication. So few examples, for example, phthalates. So you can see diethylphthalate, it's not inhibiting cell-cell communication. Neither does monobutyl phthalate, but dibutyl phthalate is a very potent inhibitor of cell-cell communication. Fluorinated fatty acids, 5-carbon fatty acid is not able to inhibit cell-cell communication. 8-carbon perfluorooctanoic acid is again inhibiting cell-cell communication. Similar situation, polychlorinated biphenyls, this coplanar dioxin-like PCB126 is not inhibiting cell-cell communication. On the other hand, non-coplanar 153 potent inhibitor of cell-cell communication. So in the next slides, I will focus more on this type of environmental contaminants. These are polycyclic aromatic hydrocarbons. So you can see that again there are some structure-specific differences in their effects on cell-cell communication. So PAHs, they are ubiquitous environmental contaminants. They are products of incomplete combustion. They are present in cigarette smoke. They are produced by fuel combustion or any type of combustive process. They are also contained in grilled foods. So they are very environmentally very so in the effects of biological activity of PAHs, we can see really structure dependent differences. So high molecular weight PAHs, which are formed from 5 or more benzene rings, they have been found that they might be highly mutagenic or genotoxic, and they also activate AHR receptor. On the other hand, these high molecular weight PAHs are less abundant in the environment than these lower molecular weight PAHs, which sometimes, like in the second-hand cigarette smoke, represent up to 95% of the total PAHs content. And these compounds, they are weakly mutagenic, less genotoxic, and they have low AHR activity usually. But when we looked into their effect on cell-cell communication, so we found that they are actually very potent inhibitors of cell-cell communication in contrast to nearly no effects of these higher molecular weight PAHs. So basically, evaluation of cell-cell communication can give us information about new chemical hazards. So the compounds which were not so intensively studied because of lack of their effects on these classical toxicological endpoints, they may actually work through some other mechanism and contribute to adverse effects on human health. In vitro evaluation of cell-cell communication can be actually utilized to provide further mechanistic understanding of the chemicals effects. So in this study, we continued with these lower molecular weight PAHs, and we chose these two representatives. Actually, it's the opposite. So this is 1-methylanthracene, which is a potent inhibitor of cell-cell communication. On the other hand, 2-methylanthracene is inactive isomer and does not induce any inhibition of cell communication. Since we knew that MAP kinases are heavily involved in regulation of cell communication, so we also studied effects of these two chemicals on two major MAP kinases, ERK and P38, and again we found that 1-methylanthracene but not 2-methylanthracene are able to activate these signal transduction enzymes, and also 1-methylanthracene but not 2-methylanthracene induced some specific changes in connexin internalization and phosphorylation pattern. So we were wondering if 1-methylanthracene activates MAP kinases, ERK and P38, whether these are responsible for downregulation of cell-cell communication in response to exposure to 1-methylanthracene. So we did these studies with a panel of pharmacological or biochemical inhibitors of different signaling pathways. And actually we found out that MAP kinases, although they are activated by 1-methylanthracene, they are not responsible for the dysregulation of cell-cell communication. On the other hand, we found that this process, inhibition of cell-cell communication in response to 1-methylanthracene, can be prevented by inhibitors of phospholipases or protein kinase A. So that kind of indicated that these enzymes are actually activated by 1-methylanthracene. So this has been confirmed by specific biochemical assays, and we found that really 1-methylanthracene activates this phosphatidylcholine specific phospholipase C, and also activates phospholipase A2, which was measured as a release of arachidonic acid. Regarding PK activation, we found that actually inhibition of these signal transduction enzymes was able to completely prevent not only inhibition of cell-cell communication, but also activation of these phospholipases and MAP kinases. So we kind of hypothesized that the PK is the upstream regulator of these signaling events. Furthermore, we decided to use some exploratory or omic approach to further characterize the mechanism which might be responsible for the inhibition of cell-cell communication induced by 1-methylanthracene. So in this experiment we focus on really short exposures, 5 minutes, and changes which were induced by 1-methylanthracene in membrane proteome. And using this proteomic approach we found that among all the different proteins identified, we could see significant changes in these membrane-associated proteins, which are called annexins. And we have seen a decrease of these annexin A1, A3, and A5 from the membrane proteome very quickly after we treated the cells with 1-methylanthracene. Reciprocally, we found that the amount of these annexins increases in the cytosolic fraction. And then we also confirmed this finding using the traditional molecular biology methods like immunostaining and Western blotting. So based on all this information, we were able to come up with some hypothetical signal transduction pathway which might be or is probably activated by 1-methylanthracene, which leads to inhibition of cell-cell communication. So basically the point is that this in vitro bioassay can be used for research of molecular mechanisms of action, either using these hypothesis-based experiments or using these omics techniques. So one of the important findings of these studies was that phosphatidylcholine specific phospholipase C seems to be the critical regulator of cell-cell communication. So we conducted this survey study where we were trying to evaluate or identify the mechanism through different environmental contaminants or signal transduction modulators can inhibit gap-junctional intercell communication. So we found that growth factors like EGF, tumor-promoting 4-ester TPA or thrombin receptor activated peptide, or environmental contaminant lindan, they inhibited cell-cell communication through this classical MAP kinase ERK-based mechanism. However, we found that all these lower molecular weight PAHs, they actually inhibited cell-cell communication through this PCPLC-dependent pathway, and also many others like PFDA, perfluorodecanonic acid, I think this is DDT, this is PCB153, and also we found that endocrine disruptors like vinclozolin or methoxychlores, they are also potent inhibitors of cell-cell communication, and they inhibit this process through PCPLC-dependent mechanisms. So it seems that it's sort of common response to many environmental contaminants. However, we also found that some compounds, they are able to inhibit cell-cell communication through some mix of these two signaling pathways. And also we found some others which did not inhibit communication through neither of those two, so probably through some alternative mechanism. When I was talking about the structure-specific effects, so you may notice that this also applies to the mechanism of inhibition of cell-cell communication. So, for example, dicumyl peroxide did inhibit cell-cell communication through PCPLC-dependent mechanism, while benzoyl peroxide through some other pathway, or these three perfluorinated fatty acids, each of those inhibited cell-cell communication via different mechanism. Okay, so that was about, let's say, use of cell-cell communication for mechanistic understanding. And now I would like to briefly demonstrate that inhibition of cell-cell communication can be evaluated using these in vitro methods in tissue-specific or species-specific context. So we did the previous data were mostly done in this rodent red liver epithelial cells, WBF344. So we were interested whether we will see the same type of response when we use human liver cells. So in this case, we work with adult human liver stem cells, which were isolated at Michigan State University. And actually, we found that these cells responded to methylated anthracene in a similar way as these red liver epithelial cells. And the mechanisms of inhibition of cell-cell communication was very similar. So basically, the tumor-promoting signaling events induced by 1-methylanthracene observed in red cells were also relevant for human liver. I did mention that PAHs are products of incomplete combustion, and they are common atmospheric contaminants. So we were very much interested if we will see this type of response also when we used some lung in vitro model. So in this study we used mouse lung epithelial cells C10, and again we work with the 1 and 2 methylanthracene as active and inactive isomer. And actually we did observe sort of expected or hypothesized pattern. So 1-methylanthracene induced rapid dysregulation of cell-cell communication in lung epithelial cells, and 2-methylanthracene had no such effect. Also, the pattern or response to of MAP kinase activation was also similar. So 1-methylanthracene induced activation of MAP kinase ERK and also MAP kinase P38, which also led to evaluation of some inflammatory markers, because P38 is involved in regulation of inflammatory responses. And indeed, we found that 1-methylanthracene, but not 2-methylanthracene, induced changes in expression of genes which are responsible for production of cytokines, cyclooxygenase 2 or tumor necrotic factor alpha. So there was like a hint or link that there might be a connection between inhibition of cell-cell communication and inflammatory responses induced in lung epithelial tissue. And our colleagues at the University of Colorado, they recently published a sort of follow-up paper where they focused on, again, the same in vitro model, 1-methylanthracene, and they evaluated also effects of fluorantine and binary mix of 1-methylanthracene and fluorantine. So one of interesting observations was that the binary mix of equimolar concentrations of 1-methylanthracene and fluorantine, it induced a more potent response and stronger inhibitory effects than the single PAHs. So that indicates that there might be some synergistic type of effect between those two. Again, fluorantine as well as the binary mix were able to activate MAP kinases P38 and ERK, and they did actually some transcriptomic studies where they studied alteration of gene expression by fluorantine and binary mix of PAHs in these mouse lung epithelial cells. So, and by analysis of this transcriptomic data, it turned out that actually one of the most affected responses or genes were those involved in inflammation. So, and that was of course confirmed by PCR experiments and also functional assays analyzing cytokine release, like interleukin 6 or interleukin 8. And also other set of genes which was heavily affected by exposure to PAHs, and those inhibiting social communication were genes involved in tissue repair and remodeling, like metalloproteinases. So that actually indicates that these are basically genes and conditions which are observed in chronic respiratory diseases like COPD or lung fibrosis. So again, there seems to be a link between inhibition of social communication by lower weight molecular PAHs and inflammatory processes in the lung epithelium. In recent research conducted in our laboratories, we also looked into effects of PAHs in human bronchial epithelial cells, and again we did see a very similar pattern to mouse epithelial cells. So low molecular weight PAHs were potent inhibitors of social communication, but again there was this structure-specific pattern like anthracene or 2-methylanthracene were not active. And also we have seen that also in the human bronchial epithelial cells, these PAHs were able to induce inflammatory responses like production and release of interleukin 8. Another tissue where we were looking at the effects of PAHs on social communication is actually testicular tissue. Again, you may know that air pollution has been linked to some decrease of male reproduction, and actually testicular tissue is able to accumulate PAHs from the environmental exposures. So we were looking into effects of PAHs into these two critical cell types in male reproductive system. So these are Sertoli cells which are actually responsible for nurturing and providing a proper environment during the spermatogenesis, and also Leydig cells which are interstitial cells which are responsible for the production of testosterone. So we are looking whether these cells can be affected by exposure to lower molecular weight PAHs. And we actually found that Leydig cells, their communication was inhibited by 1-methylanthracene very effectively. But you can see that, for example, Sertoli cells, they did not respond. So really, the effects of chemicals on social communication depend on the actual context, like cell type of cell tissue species, so they cannot be easily generalized. So very different response of Leydig and Sertoli cells. And the next part or example of results of our research is focused on in vivo validation of the in vitro data. So far I was talking about the in vitro evaluation of social communication, but you might be wondering whether these effects can be observed also in organisms, model organisms exposed in vivo. So in this study we did experiments with fluorinated fatty acids, which were found these short carbon chain ones that they do not inhibit social communication in vitro. On the other hand, these fatty acids with longer chain, they were inhibiting social communication. And we actually use this incision loading dye transfer technique to evaluate social communication in the liver of exposed rats. And we found the very similar structure-specific effects. So PFOA inhibiting social communication in vitro was found to inhibit social communication in vivo, and the shorter ones, they did not induce any inhibition of social communication. and also no tumor promotion related effects. Another example, very recent data from our colleagues who are looking into these testicular in vitro models, and they focus on this prototypical endocrine disruptor methoxychlor, which has been found to inhibit cell communication in both TM4 sertoli cells as well as TM3 Leydig cells, and also in vivo exposure of rats indicated that this chemical is able to inhibit cell communication in seminiferous tubules. So these in vitro findings might really have some in vivo relevance. At the beginning of my talk, I was mentioning that this in vitro evaluation of cell communication can be also used to identify beneficial compounds. So there are a few examples of that. So, for example, we found that resveratrol, dietary phytolexin contained in red wine and other food products, has ability to prevent these environmental toxicants from inhibiting cell communication. So if you pre-treat the cells with resveratrol and then you expose them to methoxychlor, so they do not respond by dysregulation of cell communication. And we actually used this type of approach to evaluate effects of some other dietary chemopreventive agents, and we found that really most of them were able, for example, to prevent inhibition of cell communication induced by environmental contaminant lindane. So this way, this assay can be actually used to screen for the compounds which might be beneficial for our health and can provide some kind of chemopreventive or chemoprotective activity. Another approach which can involve evaluation of cell communication is represented on this slide. So, as I said, normal cells in our bodies, they usually have functional cell communication. On the other hand, cells which have been neoplastically transformed, they have inhibited cell communication. So you can see a difference between normal red liver epithelial cells and the same cells which have been transformed by HRAS oncogenes. So these cells communicate and these have inhibited communication. And more than that, these cells are totally non-tumorigenic in vivo if you put them back to the parental organism, Fisher 344 rat. So they differentiate into hepatocyte, or if you inject them into cardiac muscle, so they form functional cardiomyocytes. On the other hand, these transformed cells, if you put them back into in vivo situation, so they start forming metastasizing tumors. So these are transformed cells. So, and what we have found that these non-communicating transformed cells, if they are exposed to certain chemicals, they can actually restore cell communication, and in connection to that also they start losing their tumorigenic properties. So examples here include sodium butyrate, silibinin or metformin, which is I think anti-diabetic drug which has been prescribed to treat diabetes for I think several decades, and let's say by coincidence it was found that the patients who took metformin, they had much lower incidence of cancer. So in this way it was discovered that this compound may have chemopreventive effects, and this kind of may indicate one of the possible mechanisms which might be responsible for that. Also, this evaluation of cell communication in oncogene transformed cell lines can be used in, say, bioassay guided fractionation to identify the responsible or pharmaceutically active compounds in complex extracts. For example, this study with cerium extract found that cerium really increased cell communication in WBRAS cells, and using this simple bioassay, these authors were able to identify the compound which was responsible for this effect, and it turned out to be beta-cytosterol. So it can be used in future discoveries. More so that in vitro evaluation of cell-cell communication is actually compatible or adaptable for high throughput screening. So there are a few examples of recent papers reporting different types of setups, how to evaluate cell-cell communication in or using high throughput systems based on principally different techniques. As I mentioned, that in our lab we use this simple scalpel loading light transfer assay. So you may wonder how this could be automatized or miniaturized. So we sort of alluded to that in these two recent methodic publications. That in fact it can be done also in microtiter plates, which then allows use of automatic image acquisition to do automatic documentation of cell-cell communication. And also for that, if you start acquiring large amounts of data, so then you need also proper methods how to analyze these large number of images. So again, our Ph.D. students are involved in this image analysis task or research. So we have come up with some tools which allow to automatically quantify the cell communication. We also introduced into this simple one-color assay two additional dyes, which allow to monitor for the dead cells and for the total number of cells. So in combination, we have semi-high throughput assay which is able to simultaneously evaluate the cell-cell communication, cytotoxic effects, or changes in cell proliferation. So that could actually be more useful for any future in vitro screening also. Okay, so conclusions. In this lecture, I was trying to argue or present that in vitro evaluation of cell-cell communication is a useful biomarker which can indicate disruption of important biological processes in mammalian tissues, and that can be used to identify either new chemical hazards or pharmaceutically relevant compounds or treatments. In addition, I was talking about new drugs or pharmaceuticals, but we use this cell-cell communication assay also to evaluate effects of treatments with nanosecond pulse electric fields or some biomedically relevant nanotechnologies or applications. This assay can be also used to characterize different molecular mechanisms of responses induced by chemicals. This can be done in species-specific and tissue-specific context, and this assay is compatible with high throughput setup as well as with in vivo validation of these in vitro findings. So there are some future outlooks where we would like to focus very soon. So we would like to further increase the throughput of our assay. So we have the targeting robot here, and now new citation 5 with BioSpot, so that actually increases the options for very high content analysis using cell-cell communication. For that, we would like to further combine this assay with evaluation of other endpoints using some additional fluorophores. We would like definitely to develop or optimize methods for evaluation of cell-cell communication in our 3D cultures. From the toxicological point of view, we are focusing on linking the effects we see on cell-cell communication as it is disrupted by environmental toxicants. So we would like to link them to other phenotypic endpoints or responses and mode of actions like liver tumor promotion, lung inflammation, or endocrine disruption within the testicular tissue. And of course, we would like to use 3D cultures to validate again these effects we see so far in traditional monolayer cultures. And we would like to focus also to further use this assay to study or identify pharmaceutically relevant compounds, like in collaboration with our colleagues from ISPA, Italy, extracts or bioactive compounds produced by jellyfish cotyloids. We have collaboration with Algatech, Academy of Sciences, to study chemopreventive or anti-cancer compounds produced by benthic cyanobacteria. And we have also collaboration with veterinary pharmaceutical university, where we are focusing on studying some compounds produced by terrestrial plants. So that's about the future. And yeah, I also asked because this habilitation procedure is not only about research but also about some contribution to the education. So how is all this research connected to education? So as Professor Klanova mentioned, that I am regularly supervising bachelor, master, and also Ph.D. thesis, and a lot of them is actually focused on research or experiments which involve evaluation of cell-cell communication. I would like to say that these theses, they involve not only students of ecotoxicology or environmental chemistry, but also biochemistry, analytical biochemistry. We were able to attract also some students from veterinary pharmaceutical university, student from University of Greifswald who was doing her master thesis in our laboratories, and also in collaboration with Algatech. I'm co-supervising one thesis, PhD thesis, which is also partially realized in our labs. Education. So there are some lectures which actually cover this topic in our class of modern methods in ecotoxicology. There is also a laboratory exercise for students which is focusing on evaluation of social communication. Within the ongoing RNDr project, this laboratory exercise is being updated to really provide the students feeling of or get some idea about these automated methods for image acquisition and image analysis. And we would like to use these elements also in a newly prepared subject or class under the new accreditation. Also, I was giving lectures to the students like in the introductory classes of special biology, and more or less regularly I speak to the students also at these events, seminar in ecotoxicology and so on. In addition to the college students, we actually welcome in our labs also students from high school or undergraduates. So thanks to very active PhD students in our group, we participate in this T Excursa project. So there is also laboratory exercise on cell communication. There are different interns who are actually starting to work in our labs on projects either during the first or second year of their college studies, or some of them are again under supervision of our PhD students, actually at the high school. And there have been also some popularization lectures and activities. So that's about the links between research and education. And at the end, I would like to thank all of you for attention, and also all my colleagues and collaborators and students from Resetox, also from Institute of Botany and from Michigan State University. and I decided to choose these three individuals because, as Jana already mentioned at the beginning, Luděk and Blahoš were at the very beginning of my scientific career as supervisors of my masters and masters and then Ph.D. studies, and also I put Clara here because we collaborated a lot during PhD, my PhD research as well as nowadays, and if I'm talking about social communication, so I have to specifically mention these three guys, so Professor Brett Upham, Professor Jim Tosco, and Professor C.C. Cheng from Michigan State University, because they had really great influence regarding the direction of my research career and further steps. And last but not least, I would like to thank our Secantox research group and all the members and associates. Sorry if I did not include somebody, but that's it. So thank you for your attention.
Thank you, Pavel. So floor is now open for questions. Clara?
Well, thank you very much for this nice overview of your research. Since you have demonstrated a couple of times that very minor differences in the structure actually make a big difference, in the effect, were there any efforts to actually identify the structural features, that are driving these differences?
So the question is, what might be the structural features which are underlining the differences in the chemical effects on cell-cell communication. So actually there have been some attempts, but I think so far it's still not clarified. So like some kind of key and log mechanism which we sort of hypothesize about, but it has not been really verified or identified. So that's still a challenge for the future research.
Well, it looks very nicely, but could you highlight it or mention some limitation where in vitro gap junctions, intracellular communication, doesn't correspond with in vivo observed effects?
So I think, okay, the question is what might be the differences between the effects induced in vitro and the effects observed in vivo. And yeah, I think there are several examples of that. For example, I think that phenobarbital, which is a known liver tumor promoter in rodent in vivo assays, when we expose our WB cells, so we don't see any inhibition of cell-cell communication. So that's kind of not consistent with our data, but we actually think that the explanation might be really in the tissue or cell type specific responses. For example, what we use, already mentioned, the WB cells, so it's a very specific cell type which exists within the liver. These are kind of stem-like cells which are playing an important role in tissue or liver tissue regeneration and repair. But it's possible that phenobarbital may simply induce liver tumor promotion in vivo through some other mechanism, not targeting these specific population of cells. So I think there are these differences for sure, but they actually can highlight really the limitations of our essays or the explanatory framework. So.
Also, you have like shown the difference. I was just still puzzled by the differences, like 1-methyl anthracene and 2-methyl anthracene, and really like different things. Is there anything in relation to the in vivo? You know, do they also show differences in vivo effects when there's such strong difference in vitro? Or is it no?
So maybe I will get back to your first question because you asked about these structural features. So actually, I need to correct myself. So we didn't find, or I think anybody has really molecular target of this 1-methylanthracene and what might be the structural difference. But actually within these, let's say, low molecular weight pHs, we can sort of see some relationship between the structure. So the presence of, we call it also Bay or Bay-like region in the molecule seems to be critical for the activity towards inhibition of cell-cell communication. So if you recall the structure of 2-methylanthracene, so it's sort of linear versus the 1-methylanthracene has this methyl group, and the same applies for anthracene versus phenanthrene. So if that molecule is kind of twisted or poses this structure features, so it has tendency to inhibit cell-cell communication. In vivo and the question was whether these structure-specific differences are observed also in vivo. Yes, I would say they are, although I think not really with 1-methylanthracene, because that I do not recall any in vivo studies with those two. But
Thank you for your lecture. I have one practical question. How will you cut and evaluate this fluorescent dye transfer in 3D spheroids? It's hard to imagine for me.
Yeah, that's a very good question, and we have been thinking about it a lot, And so, for example, there is one technique which is called microinjection. So you could, and actually for that you use a very fine instrument, so you can really locate and penetrate one single cell and inject the fluorescent dye into it and then see the dye transfer. So this could work in 3D. But we are actually thinking about a different approach, because there are two techniques which are sort of not dependent on any, let's say, invasive introduction of fluorescent dye into the cells, and they are called FREB or LAMP. So the FREB is known or widely used in life science research. It's fluorescent recovery after photobleaching. So basically, in this type of setup, you preload all the cells with some fluorescent dye. So all the cells fluoresce, and then you can use a laser beam or just concentrated So I just concentrated beam of light, and you can basically photo bleach the dye in a certain area of your sample, so that can be even a 3D structure. And then you can monitor in time how quickly the fluorescent dye from the unbleached areas will get back into the photo bleach region. So that could work, I think, with pretty much any 3D system. And LAMP is basically similar to FREB, but it works the other way around. So you preload the cells with non-fluorescent dye, and you just activate it by the beam of light in a certain place, and then you can follow the dye transfer from this region. So this is what we are thinking about that could be used in our spheroid culture. So.
My question was almost the same. On the same topic. It's been answered.
So, any other questions? If no, but
Maybe one provocative question. You talk about chemoprotective mechanisms. What's your maybe psychological point of view? Those chemicals are useful for organisms because prevent the problematic reaction, or they are toxic or negative, yeah, toxic because they fails the signaling.
Okay. Yeah, I think it can be answered like globally, or I think it really depends.
Let's say curcumin or respirator.
Mhm.
Yeah.
So, the question is whether we will do us a benefit if we sort of overregulate the social communication, for example, by exposure to resveratrol, or whether it may actually have the opposite effect, that the cells will communicate too much when they shouldn't. And yeah, so that's actually a possibility. But I think again these beneficial effects of these dietary compounds they have been actually demonstrated through like real populations and epidemiological research. So it looks like that the people who have a diet which is rich in these compounds, they usually have less health problems, or some specific types of diseases are less prevalent in them. So I think that balanced use of these compounds should be beneficial. But there is a very infamous example with that beta-carotene, which was also discovered that it has to have beneficial effects or anti-cancer effects, and actually it turned out that when people started to use like higher doses of beta-carotene, so it caused the opposite. So it's about the balance.
Many other philosophical questions.
So don't hesitate to drink wine with resveratrol.
Yeah.
But don't eat too much food.
It's always a dose.
Yeah.
Okay, we explored all the possible questions, then I just thank Pavel again for everything.